reason things are this way is how they always been and they intend to stay. I can't explain why we live this way. We do it every day.
Hey, welcome back everybody. Today we're going to take a look at the different types of mutations and their effects on a protein. So there's a couple of important things that we need to take a look at when it comes to mutations. First off, a mutation is a change in the genetic material, more specifically in the nucleotides within DNA and RNA. There are two types of gene mutations. There's point mutations and frameshift mutations. Let's go over some point mutations, and really there's a change being made in one nucleotide. And this type of point mutation is called a substitution. And it's when one base pair is changed into another. So in our normal DNA, T and A complement each other, but in the mutation, instead of having the A there, we're going to have a C, and that's going to make change to one amino acid. We learned in the previous lesson that every 3 nucleotides equals 1 amino acid, and since it's only affecting one nucleotide, it will only affect one amino acid. Frameshift mutations, on the other hand, are a little bit more complex because it's not just affecting one amino acid, it's going to affect all the amino acids after that mutation. So a mutation that shifts the reading frame of the genetic message by adding or deleting a nucleotide is a frameshift mutation. So let's take a look at a deletion. Down here we have our normal DNA coding strand, and through transcription we have the messenger RNA, and then during translation we get these amino acids in a sequence. However, over here, if we take that same DNA coding strand and we just delete one of the A's, we had 3 here and we only have 2 here, and we delete that A, look and see what happens to the amino acid chain. The first 2 are still the same because the deletion happened after them, but for every amino acid after, there are different amino acids being placed in that chain. This can strongly influence the productivity of that protein. It can influence how well that protein works, or if it even works at all. So mutations can be a big deal. Other mutations aren't such a big deal. On the previous slide I kind of hinted at the significance of mutations, but there's really 3 types. There's neutral, there's negative, and then there's beneficial mutations. The first type, neutral, that's what many of the mutations are. There's little or no effect on the expression of genes or their function. The second is a mutation that is negative because it can cause dramatic change in the protein structure or the shape, which will disrupt normal biological activities. These types of mutations can cause disorders, which we'll take a look at in the next chapter. And the last one is beneficial mutations, and often animal breeders and farmers take advantage of this type of mutation. For example, sometimes we like to cross dog breeds, and we like to introduce a new characteristic or a new gene to specific dogs, and we can do this by controlling a mutation or taking one mutation from one dog and putting it into another dog. For farmers, no one likes watermelon with seeds in them unless you like to spit them in the summertime, but some people enjoy seedless watermelon. This was made by a mutation or some type of gene therapy that would allow this to happen. So today we went over point mutations and frameshift mutations. Point mutation, think if you point at one thing, you're looking at one amino acid change, where frameshift mutations will affect all the amino acids that come after that mutation. If you have any questions on mutations, please don't hesitate to ask them in class.